So, good morning, everyone. I think everyone has joined by now, and the rest who has not, they might join us later. So, in today's lecture, yesterday we have discovered about various biomolecules, how they are important from the biochemistry perspective. So, now we will take each of these biomolecules one by one and discuss them. So in this lecture we will be discussing about a little bit about amino acids, how they are having these optical properties, levo and dextro, their acid properties, um, being amphoteric in nature, yeah, buffer characteristics, physical properties, and their importance and functional roles. That we will get to know about how they're acting as zwitterions and various other perspective of amino acids one by one. So amino acids, as we know, they are the building blocks of the proteins, and their general characteristics or structure of an amino acids, it includes an amino group first of all, on the left hand side, and there is a carboxylate group, alpha carboxylate group on the right hand side, and the variable side chain, that is R. Yeah, so this will be common in all of the amino acids. So here we can see that this is amino group and there is a carboxylic group. So common to all alpha amino acids of proteins. And this is alpha carbon, which is between the carboxylic and the amino acid group. And this is the side chain for each amino acid is different. And according to this, we having different properties, various characteristics of our amino acids. So because of change in this R, we have distinguished them into 20 amino acids. So these amino acids, as we can see, it has positive and negative, both sides. This is positive and this is negative. So as the amino acids contains ionizable groups, so they can act as an acid as well as base in a solution depending on the pH of the solution. So due to the nature of amino acids, they are called as zwitterions or dipolar ions, right? They are called as zwitterions or dipolar ions because of having both nature of acidic and basic depending on the pH of the solution, and this is happening because of the ionizable group, that is your plus NH3 and minus COH. So they do not have charge but behave as acid and base depending upon the pH changes in the solution. So in usually they are neutral, but as the pH changes, so they started to act as a base or acid. So the biological importance of amino acids. So there are over 200 different amino acids that occurs in nature, but only 1 of 10th these occurs in proteins. They are mainly known for making proteins, but actually in nature they are 200, but out of them 20 amino acids they are meant to make protein. And the biological functions that they performed by them are, so as we know, they are the building blocks of peptides and proteins, and as precursors, many biological important molecules are derivatives of amino acids. For example, let's say tyrosine, that's amino acid, which is a precursor of the hormone thyroxine and a skin pigment melanin. So tyrosine is a precursor of a compound abbreviated as DOPA, dihydroxyphenylalanine. So tyrosine has been a very essential amino acids in the production of your thyroxine, melanin and DOPA. which having a very essential role in our body. So they are also acting as a neurotransmitters, that is the transmission of impulses in the nerve system. So tryptophan is a precursor of vitamin named a nicotinic acid, like vitamin B3. Yeah, so in the case of like transferring from the neuro generative signals, in that case also these amino acids playing very essential role. Also in the case of vitamins, they are also acting as a precursors. Then there are also sources of sulfur, so derived from the sulfur containing amino acids like cysteine, right? And beyond that, they're also involved in many metabolic pathways such as gluconeogenesis, where it is involved in the glucose synthesis. Yeah, so these are the, so we will discuss about glycolysis, gluconeogenesis, all these pathways in upcoming lectures. So in that case also our amino acids playing a quite essential role. Then physical properties. What are the various physical properties of amino acids? First of all, so they are the mainly water soluble, that explain its polarity and the presence of the charge group. So they are soluble thus in polar solvents and not soluble in the non-polar solvents. Because of polarity, they have this property, and that's why they are able to dissolve in the polar compounds but not in the non-polar solvents. And they tend to have very high melting point. Yeah, um, like to break the bonds of these ionic forces, and they maintain a very strong crystal lattice of the protein, so it's very hard to break that. So that's why the melting point could be sometimes above 200 degrees Celsius. Yeah, boiling point is if it is 100 degrees Celsius, then it's a double that amount, 100 times. And in nature they are colorless crystalline substance which are soluble in water. Then comes the optical activity. So it is exhibited by all amino acids except for glycine. It does not have asymmetric carbon. Only the glycine is the exception here. So remember this part. Glycine is the one which does not have asymmetric carbon atom, so it does not show any optical activity. No. So there are two types of optical activity: dextrorotatory, levorotatory. Yeah, the dextrorotatory are the one which has ability to rotate the plane of polarized light to the right. So if we put them in water or any mixture and the light is coming, if we put the light towards that single light, so it will move that light towards the right plane. And the laboratory will be the those amino acids which have ability to move your polarized plane of polarized light to the left side. So both can rotate the plane of polarized light with the same magnitude but in the opposite direction. Then acid-base properties of amino acids. So all amino acids contain at least two ionizable groups. As we have discussed, they're having two ionizable groups, that is your alpha amino group and alpha carboxyl group. And some of amino acids, they also have additional acidic or basic group in their side chain. The R chain that we discussed, the bottom one, it also have additional acidic or basic group which are responsible for amino acids acidic and basic behavior. So if they are present, it's okay, they have to be present, this makes them neutral. But beyond that, into the alkyl group, your free side chain group, some more acidic or basic will be attached, and this will make them amino acids more acidic or basics. And the equilibrium that we tend to see in these amino acids, that the carboxylic group, the side chain is added, it will make H will be removed, it will become negative, and like if proton is removed, and in case proton is added, then it's become positive, your RNH3. So since these reactions are reversible, this indicates that they can act as acid or bases. So the reaction that we see here, they go up and back and forth. Anything is possible. So because of that, um, they can act as acid or bases, which explain the acid-base behavior, and thus they are also known to be having amphoteric property, having the both acidic and basic properties. That's why they are also called as amphoteric in nature. Then comes the next point, that is zwitterines. So as I said, the all neutral amino acids are present in zwitterine forms at physiological pH, that is 7.4. At 7.4 they will be all neutral, and the carboxylic group will be unprotonated and the amino group will be protonated. In this case, this is unprotonated and this is protonated. Like carboxylic group tend to lose mostly the proton, and whereas the amino group tend to gain electron. So in that case, that's why they are also known as zwitterines. Yeah, so there's two different terms which seems to be quite similar in the case because of the amino acid and carboxylic group. But remember, amphoteric property is based upon the acid-base behavior, whereas the zwitterine property is based upon the pH, where the carboxylic group and amino group are getting unprotonated and protonated. Remember the difference between two. Both terms look similar, but in the objective time sometimes come. So in the amphoteric property, they might write the properties of zwitterines, and in the case of question of zwitterines, they will write the properties of amphoteric properties. So these terms are meant to be made confused. Yeah. Then amino acids can be present in two configurations, as we have discussed: L isomer and D isomer, levorotatory, dextrorotatory, and both of them are actually the mirror images of each other. It's just they are in the different arrangement of alpha carbon atom. The alpha carbon atom, uh, and the proteins and the amino acids could be called as are in the L configuration or L amino acids. So here we can see there's L alanine and D alanine. Yeah. So if you put this image, same image in the mirror, you will see if amino group is attached here, it will see on the right hand side. So it's become dextrolovatory and this has become levolovatory. So your alkyl group is there, your carboxylic group is there, amino group is here. So just mirror image of the same. Now, as we have discussed since morning, started with the amino acids, the basic structure of amino acids, how they're acting as acid and base, their biological importance, physical properties we have started, then optical activity we have discussed, then we have discussed about the acid and base properties of amino acids, then we have discussed the amphoteric and zwitterine properties of amino acids, these L-alanine and D-alanine, how they are on the left image and right image. And now let's take to the next part of amino acids, that is your chemical properties of amino acids. So the reactions which occurs due to the presence of amino group, that is NH2 group. So first reaction, very famous reaction, Nin Hydrogen reaction. It's a very powerful oxidizing agent, Nin Hydrogen, that could decarboxylate, that is remove the carboxylic group from the amino acids, yielding CO2, NH3 and aldehyde group. CHO group, NH3 and your CO2. So it removes it's like it get decarboxylation. This is just break down your amino acids and break down into CO2, amino acid and aldehyde. And the reduced Nin hydrogen reacts with the liberated ammonia to form a purple color. So after that reaction happens, so Nin hydrogen will be reacting with the ammonia, yeah, to form purple color complex called rumen purple. So this is the like confirmation that whether in your chemical, whether in your mixture, um, is present or not your amino acids. If the purple color comes when you react with the Nin hydrogen, that means it has break down the amino acids into CO2, ammonia and aldehyde, and Nin hydrogen, the reduced Nin hydrogen then will react with the liberated ammonia to give a purple color, which is also known as Ruhmann purple. Then some another properties of amino acids. So they could be identified by specific color reactions. There is called xanthoprotic test by benzene ring of aromatic amino acids. So there are some amino acids we will be talking about. They are having these aromatic, these benzene rings um, over the side chain group, and because of that presence, we can understand that okay, this is confirmation with the xanthoprotic test. We can confirm it, and there is a Millen reaction for the presence of phenolic group um, like tyrosine, and the sulfur test is done for the sulfur hydryl group of amino acids, and Nin hydrogen test for the alpha amino acids. So we can see there are many tests are possible in order to confirm various different types of amino acids. So xanthoprotic test, Millen reaction, Myelin reaction, sorry, sulfur test, and Nin hydrogen test. So 1, 2, 3, 4. So each times when you do them, you get a different color direction. And then beyond these things, there is also carboxylic group present. They are also very important for the reaction. So let's see. So amino acids form salts with bases and esters with alcohol. So they form salts with bases and esters with alcohol. So if some base will come, it will form this CONA and the carboxylic group of the amino acids, and it will form esters group, yeah, with alcohol group also. And also decarboxylation could happen, that is removal of COH group, and amino acids undergo decarboxylation to produce a corresponding amines. So this reaction is important in living cells due to the formation of many biological important amines like histamine, tyramine from the amino acids like histidine and tyrosine. So they are very essential in the biological processes of in our body, and the source of these essential amines are these amino acids histidine and tyrosine. Mhm. So, in this short lecture about amino acids in this morning, we are able to understand how these amino acids, they are the building blocks of proteins and are the biomolecules of the life. And we have understand various physical properties and chemical properties of amino acids will help in understanding their metabolism. Yeah. So some references. Um, So yeah, I want to share today some books with you. Um, I have many biochemistry books, so will be helpful. One is Harper, one is Lippincott, and one is Lippincott is for the medicinal perspective. Harper is the basics, and also Lehinger is more advanced. So all these three books has its own characteristics, own importance. So use them wisely. Yeah. So whatever we are discussing here today, it's all present within the content. Yeah, metabolism, intermediate metabolism, lipid metabolism, nitrogen metabolism, integration of metabolism. So basically, it has 6 units. Yeah, so quite essential book. So this is the content from the Lippincott and Harper. This is more like protein perspective. If you see the contents of it, so they're around, wow, it's 54 chapters there. So it's include all the, it has covered most of the your basics of your biochemistry. You can go for kinetics, acid basic properties, your citric acid cycle, glycolysis, glycogen, no? Fatty acids, nucleotides, metabolisms, um, hormone action and signal transduction. This is from the medicinal perspective, muscle and cytoskeleton, right? Hemostasis and thrombosis, right? And vertebrate. So it's more actually inclined to your medicinal perspective, this Harper book. Okay? And whereas the third book, that is your Lehinger, it's a broad. It has everything. So this is also divided into around 28 chapters, but this is very much defined. I mean, this is not more inclined to your medicinal perspective. There might be some chapters related to that, like let's say this one, hormonal regulation integration of mammalian metabolism. But the content in the Lehenger, the way they have presented, it's more easy to understand and very like diagrams are very nice in it as compared to the other books. So in that perspective, I will really prefer this book out of the others too. So so now it's like these are the international authors books, all three, and this is highly recommended if you have to study these biochemistry subjects, medical biochemistry, medicinal biochemistry. So they are very well known for the same. Yeah. So happy learning with them. Okay. Now next part. Classification of amino acids. Mhm. So in this structural classification of amino acids, so we'll be discussing about various classes of amino acids, various disciplines within it. You as a disciplines, you will learn about that, and also we will understand different types of structures and their role with aspect to amino acid. Okay. So amino acids, they are the molecules which contain an amine group and a carboxylic acid group, as we have discussed, and a side chain which is specific to each amino acid. And the key element of these amino acids are carbon, hydrogen, oxygen, and nitrogen. So these amino acids are the basic structural building units of protein and other biomolecules, and they are also utilized as an energy source. There are essential and non-essential amino acids. Uh, so as we have discussed, there are in total 20 different amino acids that make up all proteins in the human body. So these amino acids are needed to replenish tissue, red blood cells, enzymes, and other substances. So 9 to 12 can be manufactured by the body, non-essential amino acids, and they are not obtained from the diet. Yeah, and the remaining 8 to 12 essential amino acids, which are not produced by the body, must be obtained from outside the diet. So, so these are the names of the various non-essential amino acids, that is alanine, asparagine. You should remember this name. I mean, they are very essential to remember. Actually, you can remember them as I have made this thing in order. So alanine. So first A4, 4 times A, 4A. Alanine, asparagine, aspartic acid, arginine. Then C, then glutamine, glycine, glutamic acids, 3G. Then serine, proline, tyrosine, SPT. And in the case of essential amino acids, isoleucine, then leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So in this way, your non-essential and essential series will be completed. So these essentials we required from our diet, outside diet, whereas a non-essential, they are manufactured, these are manufactured in our body, all of them, so we do not require them from outside diet. So some functional classification of these amino acids, like these 20 amino acids, how they are being grouped together. So as we have discussed yesterday, there's anabolic and catabolic responses and tissue pH regulation. So in order to maintain that, we need your glutamic acid and glutamine. And to maintain the urea and nitrogen cycle, to maintain those cycles, so we need these 3 amino acids: arginine, aspartic acid, and asparagine. And essential amino acids for proteins and energy. So these are isoleucine, leucine, valine, threonine, histidine, and lysine. So these are required for our body. And sulfur containing amino acids for methylation and glutathione, they are your methionine and cysteine, only 2 amino acids. Further, neurotransmitters and precursors from the brain perspective: phenylalanine, tyrosine, and tryptophan. So these are the 3 amino acids, that is your phenylalanine, tyrosine, and tryptophan. Further, precursors to heme, nucleotides, and cells. So for the membrane proteins, it will be glycine, serine, alanine. And for bone collagen specific amino acids, they are proline. So these are the various characterization of all these 20 amino acids according to the functions has been characterized in this order. So neurotransmitter is phenylalanine, tyrosine, tryptophan. For heme, it's glycine, serine, and alanine. For bone collagen, it's proline. Yeah. For essential for the protein and energy, these are iso, leucine, valine, threonine, histidine, lysine. For methylation and glutathione, it's methionine and cysteine. And for the urea nitrogen management, it's arginine, aspartic, and aspargine. And on the other case, for the tissue pH regulation and anabolic catabolic responses, glutamic and glutamine. Yeah. So this is the chemical structure of various amino acids. This we should also remember them from the examination and actually for the general knowledge perspective. So here we can see all 20 amino acids, right? They have been divided into 4 different parts. One is non-polar, polar, positive charge and negative charge, right? So let's see first the positive and negative ones. So here these are lysine K, the short. Remember this also alphabets, the short alphabets. K and R. Arginine is with the R, and histidine is H. So they having the positive charge. Why? Because of the in the alkyl group here they have the in the side chain group they have this ammonia present, NH3. Here also NH2 plus. That's why they are positive, NH plus. Okay? And the negative charge, they are aspartic and glutamic acid. Aspartic is with the D and glutamic with the E. So remember these exceptions, because sometimes for the lysine we will type the short form as L, because these are essential if you want to study about proteomics in your future, or mass spectrometry or HPLC. So in that, whenever you will get the readings, you will see the amino acid chains value coming not in the full form like lysine, arginine. So in that you have to read your amino acids in the form of K, R, H, D, yeah, E. So we should know that D is aspartic acid, E is your glutamic acid, and Q is your glutamine, and so on. So these letters are really essential to remember. So some are with the same, but some are not actually. So in this case, in the positive and negative charge here, only the histidine has H, and the rest, lysine is K, arginine is R, aspartic is D, and glutamic is E. And they are negatively charged because of this carboxylic group, O negative, O negative. So these are positive and negative. Now comes polar and non-polar. Polar will be the one which will be dissolved in the water. So these are with the OH groups. So serine, threonine, yeah, tyrosine, asparagine, and glutamine. So glutamine is Q, asparagine is N, tyrosine is Y, threonine is T, and serine is S. So this only these two have the right name S and T. The rest, tyrosine is Y, asparagine is N, and glutamine is Q. And these are having these following structures they are present. This picture that we are seeing is very essential and easy to remember the structures also. And for the non-polar, this is the most common structure is glycine. Then you have alkyl group attached to it, alanine. Then 3 alkyl group, then valine. Then sulfur group if it attached to the alkyl group, cysteine. Then making this pentagon, this is proline. This is 4 alkyl groups, then leucine, and 1, 2, 3, then 5 alkyl group isoleucine, and methionine in between. This is sulfur group here. Yeah. Then tryptophan, this your benzene ring and 5 pentagon ring, and then phenylalanine also having these aromatic rings there. Yeah. So that's how your non-polar, polar, positive charge and negative charge, your whole 20 amino acids are divided in this way. Just try to draw this diagram in your house and remember these names. So it will be very helpful on the long term. Yeah, I hope all is going fine there. You're all present. Okay, good. So their uses in human nutrition. So when we taken up into the human body from the diet, so there are 22 standard amino acids that are used to synthesize proteins, and other biomolecules are oxidized to urea and carbon dioxide as source of energy. Further, the oxidation pathway starts with the removal of amino group by transaminase, and the amino group is then fed into the urea cycle. So the other product of transamidation is a keto acid that enters the citric acid cycle. Then glucogenic amino acids can also be converted into glucose through gluconeogenesis. And some non-protein functions. So these amino acids are used to synthesize other molecules. For example, tryptophan is a precursor of neurotransmitter serotonin, and tyrosine is a precursor of neurotransmitter dopamine, and glycine is a precursor of porphyrins as heme, and arginine is a precursor of nitric oxide, and aspartate, glycine, and glutamine are the precursors of the nucleotides. So, all in all, we have acquainted with the basics classifications of amino acids, and we have understand their basic structures too, how their basic structures works like. So, Lippincott, Lehinger, Murray, Mark essentials. Think about it. So, this was the basics of classification of amino acids. So, I think I will stop it here today, because if I start with another topic, I want to start with lipids now in details. Yeah. Students, I also have some from AKTU. It's Dr. APG Kalam, Abdul Kalam University. These biochemistry complete notes. So lipid metabolism, amino acid metabolism, nucleic acid metabolism, enzyme. Yeah. So it will be good for you to have a short notes regarding the biochemistry. So they are quite good actually, made from various books and sources. So let me share this also. Okay, on that note, we are done for today, and then we see us tomorrow at the same time with more discussion about lipids and carbohydrates. So how lipids and carbohydrate works. So we will study about them tomorrow. So thank you very much for coming up in early today morning. So being part of the session. Gradually we go to the more advanced topics in upcoming days. Yeah, wish you a nice day, working day. I hope I don't wake you up so early, but it's good you are awake, and you can manage your many things in upcoming day then. Till that time, till tomorrow, take care. Bye bye.